Assalamu alaikum. So in this lecture we'll talk about the nucleus. Now you know what the nucleus is. It's basically a repository of genetic information. It contains our genetic material, our DNA. Now what's important about the nucleus is that it is enclosed by a membrane. Okay, so it's called the nuclear envelope right here. And DNA, by the way, is not arranged randomly inside the nucleus. Rather, it is organized in a certain way, as we will see in this lecture. Okay, so the thing is that's the nucleus is enclosed, it is surrounded by a double membrane structure called the nuclear envelope. But before talking about the nuclear envelope, we'll talk about the matrix of protein. So the nucleus is actually organized or protected in a certain way by a network of proteins known as forming a nuclear lamina. So it is the cytoskeleton of the nucleus. Okay, we'll talk about these proteins in a few seconds. Now, so it's a double membrane structure. The nuclear envelope is a double membrane structure. There is an outer membrane and there is an inner membrane. Between them you have the lumen, okay, which is very similar to the ER lumen, because the outer membrane is actually a continuation of the endoplasmic reticulum, except that the proteins that are on the surface of the outer membrane are different than what's on the ER membrane. Okay, they both also have ribosomes on the surface. Now, the inner membrane is unique, and it binds to the lamina, the nuclear lamina, the cytoskeleton. Now, in the membrane, in the nuclear envelope, you have the pores right here. You have the nuclear pores, and we'll talk about them as well, because they organize and they control what goes in and what goes out. Now, so the nuclear lamina is actually made of three genes, and these three genes can code for 7 different proteins. They are intermediate filament proteins. Okay, so we'll talk about the cytoskeletal proteins next time, and there are different types of proteins. Okay, so one of them is known as the intermediate filament. Okay, and basically what you have is that you have the lamin protein. So you have 3 genes: lamin A, lamin B, lamin C. They form a chain and then they form a dimer. This dimer can form more organized structure and complex structure, and all of them are connected to each other head to head. Okay, so and then side by side. Now, what's important about the nuclear envelope is that it interacts with the lamina. Okay, and there are proteins that are present in the envelope that interact with the lamina, and proteins that can also interact with the cytoskeletal proteins. Okay, so you have proteins right here, emerin and LBR, and these proteins can interact with the chromatin. Okay, so right here in yellow you have the lamina, and this lamina can interact with the chromatin. Okay, and they can also interact with proteins on the nuclear envelope. And this is really important, because you can also have these proteins interacting not only with the chromatin, but they can also interact with the cytoskeletal filaments in the cytoplasm. And these cytoskeletal filaments can interact with the plasma membrane. So you can think of the signal that is transmitted from outside of the cell that would change the structure of the cytosolic cytoskeleton, which and this change can be transmitted, or the effect of this change can be transmitted via these proteins to the lamina. Okay, and since lamina can interact with the chromatin, you can see the effect. So the change would be transmitted from outside of the cell all the way to the DNA via interaction. So it's almost like this game that we used to play when we were young, when we used to talk to each other using two cans and a rope in between. So it's the same idea. So a signal can be transmitted via this rope. So you have what is known as a link complex connecting the nuclear lamina with the cytoskeleton outside of the nucleus in the cytosol. And you can have another complex, the lamina-chromatin interaction, whereby the lamina can interact with DNA binding proteins, whether they are histones or non-histone proteins. And what's really interesting, and we'll talk about it at the end, is that remember how we talked in the molecular biology part that you have heterochromatin and euchromatin. So euchromatin contains the active genes, and heterochromatin is composed of inactive genes. If you look at the organization of these genes, you would find that heterochromatin is found in the periphery of the nucleus. And the euchromatin that contains the active genes, they are inside the nucleus. Okay? We'll talk about this at the end. So, any defect in the laminin proteins can be pathologic. Okay? So you have different conditions that are associated with mutations in these proteins. So like what is known as X-linked Emery-Dreifuss muscular dystrophy. It's caused by mutation in aberrin. You can also have mutations in lamins A and C causing different types of mutations as well. Okay, different forms of the Emery-Dreifuss muscular dystrophy. So you can have the same disease caused by mutations in different proteins. Not only that, but you can have also mutations in lamin A causing different diseases, like Mary-Charcot disease, Toth disease. Okay, it's a common disease that results in muscle wasting. You have the Hutchinson-Gilford progeria, which causes premature aging, as you can see here. So you can see a little child looking like an old man. And there is the Donegan type partial lipodystrophy. And the thing is, how is it that you have one gene causing many diseases? Okay. So and we'll talk about this further in the genetics part. But the idea is that what happens is that the interaction between lamins and the chromatin, lamin and DNA, might result in changes of gene expression. So these mutations would cause activation of certain genes and inactivation of other genes, resulting in depending on the mutation, in abnormality in the expression of certain genes that would cause one disease or the other. Okay. And the other theory is that there is something known as a mechanical stress, meaning that, especially in muscle cells, the connection between the cytoskeleton with the lamina, okay, would cause abnormalities in muscle cell function. Okay, so it has nothing to do with gene expression. Rather, it's the interaction, it's the shape of the cell, its ability to resist stress on muscle cells. Now, we said that the envelope contains nuclear pores, and these pores control what goes in, what goes out. So they're composed of different proteins. They have a specific structure, but one of the things that are important about pores is that they contain proteins known as FG-NOPs. Now, what does that stand for? F stands for phenylalanine. G stands for glycine. So these proteins are rich with these two amino acids, phenylalanine and glycine. So phenylalanine, glycine, nuclear proteins. Now, these proteins regulate what goes in, what goes out. What's important about the nuclear pores is that small molecules can pass through the nuclear pores via passive diffusion, but large proteins like RNA molecules as well as proteins, they are regulated. Their transport is regulated, as we will see. So, how do nuclear proteins get into the nucleus? There is a nuclear localization sequence, and in fact there are two sequences. There's a sequence that is possibly charged and it is continuous, and there is another sequence that is also possibly charged but it is separated, and it's known as bipartite. Okay, now these amino acids that separate the two sections or the two parts of the sequence can be mutated without affecting transport of these nuclear proteins. Okay. So how are these proteins imported? How do they get inside the nucleus? So what happens is that you have a protein with a nuclear localization sequence. It gets recognized by a protein known as important. So important binds to this protein and it takes it all the way inside the nucleus. And then what happens is that you have another protein known as a RAN. Now this RAN is bound to GTP. When it is GTP bound, it can interact with important, releasing the cargo, releasing the protein inside. Now you have the RAN protein interacting with important. Now important must get out of the nucleus so that it can function again. So RAN takes it outside. Once it takes it outside, you have a protein right here associated with the nuclear pore. It hydrolyzes GTP to GDP on RAN, releasing the important protein so that it can bind to another protein with a nuclear localization sequence. Okay, so now RAN is bound to GDP. It can bind to another protein regardless of the name of that protein, and it takes it inside the nucleus again so that it can exchange GDP to GTP, and now it's active again. So RAN is active again, so it can bind to another important, releasing the cargo and so on. Okay, so that's the idea. Important binds to the nuclear protein, it takes it inside. Once it's inside, it interacts with a protein called RAN when it is GTP bound, releasing the cargo protein. Important and RAN, both of them go outside of the nucleus. Once they get outside, GTP is hydrolyzed to GDP, so RAN dissociates from important. Important can function again. Now RAN would bind to another protein that would take it inside the nucleus again. Inside the nucleus, GDP is exchanged to GTP and it can bind to another important and so on. These proteins, and we'll talk about them a lot, by the way. RAN, RAS, RAB. We talked about RAB in vesicular transport. We'll talk about RAC and RO. These proteins are called small GTP-binding proteins. So they are, their function is dependent on binding to GTP or GDP. And they are regulated by proteins known as guanine nucleotide exchange factors, GAFs. What these do is that they catalyze the release of GDP, okay, so that GTP can bind to these proteins, RAS, RAN, RAB, whatever. Okay, so GTP comes in, GDP goes out, now it's GTP bound, now the protein is active. Now, what happens is that you have other proteins known as GTP as activity, because these proteins, the G proteins themselves, the small GTP binding proteins, have an intrinsic enzymatic activity. That is, it's inside them. The activity acts on the protein itself. It doesn't act on other proteins. It acts on itself. It hydrolyzes GTP to GDP, and this enzymatic activity is catalyzed by the GTP as activating proteins. Okay, so once you have the protein bound to GDP, it's now inactive, and you have to exchange GDP by GTP. So remember how these proteins function. So RAN also plays a role in exporting proteins, in getting proteins out of the nucleus. So how does it do that? Well, when RAN goes into the nucleus, it binds to a protein known as exportin. Okay, now this exportin so binds to RAN when it is in the GDP bound form. So RAN goes in. Once it's inside the nucleus, it releases the exportin protein. Okay, what happens is that this exportin protein can bind to the cargo protein. And the cargo protein, in association with the exportin, can associate with the RAN in the GTP bound form. And it takes it outside. Okay? So once it goes outside again, you have the GTPase activating protein, hydrolyzing GTP to GDP. Now RAN releases the cargo protein, and the cycle goes on again. So these proteins, importans and exportins, they are collectively known as karyoferins. Okay, because that's what that's their function. Okay, so importans and exportins are known as karyoferins, and these karyoferins, the function of these karyoferins is dependent on RAN proteins. So that's for exporting, importing proteins. How about RNA? Well, RNA, there are different types of RNA molecules, right? We have the mRNA, the rRNA, tRNA, and we have many other non-coding RNA molecules, and they are exported in a different mechanism. So for example, ribosomal RNA molecules are exported in association with ribosomal proteins that are found in the nucleolus. We'll talk about the nucleolus in a few seconds. Okay, and these proteins have a nuclear export signal, and they are dependent on an exportin protein known as chrom1, and their export is dependent on RAN proteins. Now, on the other hand, exporting mRNA after processing is independent of RAN. That's one, that's important. It doesn't involve karyoferins that we just talked about. They are transported through the nuclear pore complex by an exporter complex, and they are released by a helicase that's associated with the nuclear pore complex. Okay, so their export is different than all of the other RNA molecules. Now, what's fascinating is that DNA is not randomly organized in the nucleus. Rather, it's organized in a certain way whereby you have domains of DNA. So it just happens that heterochromatin, which contains inactive genes, they are associated either with the nuclear lamina, okay, or it is associated with the lamina that surround the nucleolus. So this is the nucleolus right here. So what is the nucleolus? It's a region in the nucleus that is the site of ribosome synthesis, ribosomal proteins, ribosomal formation of ribosomes, that is connecting ribosomal RNA molecules with ribosomal proteins. Okay, so that's heterochromatin, by the way. It's associated with the surface of the nucleolus or with the lamina on the periphery of the nucleus, on the outside of the nucleus. On the other hand, transcriptionally active domains are localized in the interior of the nucleus, right here in the middle. And these domains, the heterochromatins and the euchromatins, are formed by proteins known as CTCF and another protein known as cohesin. Okay, so you have these proteins that separate the heterochromatin from the euchromatin. Now, And it just turns out that there are what I call nuclear factories. Basically, there are specific regions where you have active DNA replication or active transcription going on. So you have the active genes, for example, or we'll talk about replication factories first. So what happens is that you have discrete regions inside the nucleus where you have the origins of replication found in these regions. Okay, so they all happen at the same time. So this is sort of like trying to organize replication. So these proteins that are associated with opening up the origin of replication, they all exist in the same region inside the nucleus. So it's really organized. And you have transcription factories as well. So active genes, okay, so right here you have the heterochromatin, and active genes which are located in the euchromatin part of DNA, they are localized in the interior of the nucleus, and you have all these transcriptional factors now also concentrated in these regions. So you have active genes happening in certain regions in the cell. So this is still an area of active research, by the way, and it's fascinating, by the way, how things are organized. So the nucleus is not really randomly organized. It's not like DNA is scattered all over the place. Rather, you have specific regions of the nucleus where proteins are organized, where activities are organized. So we talked about the nucleolus. Nucleolus is where you have rRNA transcription and where you have the assembly, the biogenesis, the formation of ribosomes. Okay, you have specific other types of regions inside the cell. So for example, you have things known as speckles, and speckles are regions where you have storage of pre-mRNA splicing factors. So this is where mRNA splicing takes place. Okay, these ones here. Okay, so the nucleus is not really randomly organized. Again, this is an active area of research, and we will learn more about it in a few years.